So our next speaker is from Dr. Chris Chen's lab here at BU. He's a member of the Biological Design Center, also professor of biomedical engineering. Jourdan Ewoldt is going to talk about this. Whoops, I have to reset my timer. But what's important is about the Chen lab and this project is that it's related to the Engineering Research Center that's funded by NSF here at Boston University. And the function of that center, the main goal of it, and it's just been renewed, is to create three-dimensional cardiac tissues that can be used either as a test bed to test drug interventions for cardiac and diseases, but also to actually make tissues that could potentially be used as patches in tissue engineering constructs for tissues that have been destroyed or harmed by heart attacks or other types of cardiac toxicity. So I am going to hand it over to Jourdan.
Okay, hi, I'm Jourdan. I'm a PhD student as part of CELMET, which is the National Science Foundation Engineering Research Center. So like Kathy said, it's a very large grant for about 10 years that's multi-institutional. So there are labs from BU, University of Michigan, Florida International University, Columbia, and more. And so we're all coming from different engineering and biological backgrounds with the ultimate goal to create that engineered heart patch. But as we were working towards our larger goals and our different research applications, it became evident that we need potentially to have automation to be able to scale up to actually achieve those goals. So that got Chris talking with the DAMP lab about a potential partnership. So today I'm going to talk about our pilot project that's led by Rita Chen in the DAMP lab to automate our differentiation of stem cells into the heart muscle cells called cardiomyocytes. And this is also helped out by Josh, who is part of CELMET. So there are three main applications for engineering heart tissue. The first one is to treat the heart after myocardial infarction, also known as a heart attack. So after a myocardial infarction, the heart muscle cells, cardiomyocytes, they die, and like other parts of our body, those cells don't regenerate. So the goal of the field and of CELMET is to introduce stem cells that are becoming cardiomyocytes to the heart to restore that contractile function to decrease the chances of heart failure after myocardial infarction. This is still in progress. It's a very lofty goal, but currently efforts can take up to 1 billion cells to be able to deliver to the heart to treat it. And these cells don't proliferate, so that means you have to make those billion cells per treatment. Another application is for drug screening. So currently it can cost up to $1 billion to get a pharmaceutical drug to market. However, more than 10% of the drugs are removed due to negative effects on the heart, whether that be cytotoxicity or causing arrhythmias. So if we are able to have a human in vitro study to do a high throughput screening of these drugs before getting to clinical trials, it can save a lot of money and time and help create more pharmaceuticals. And lastly, there are hundreds of different mutations in genes that encode the heart muscle cells that lead to genetic cardiac diseases called cardiomyopathies. And these ultimately lead to heart failure. So if we were able to study all these different mutations in different cell lines, we'd be able to better understand how these mutations affect the heart cells and how we could maybe make treatments for people with these diseases. So for all these applications, people are converging in the field on using induced pluripotent stem cell derived cardiomyocytes or iPSC-derived cardiomyocytes. And there are a lot of different protocols for this, but essentially the field is using iPSCs, activating the Wnt pathway over a course of time, which makes the cells become mesoderm-like, and then inhibiting that pathway to make them more like cardiac progenitor cells, and then eventually adding insulin to make them cardiomyocytes specifically. There's a lot of variability in the field. So as a center, we've been analyzing over 300 papers to really quantify that variability and to produce standards to be able to decrease this within the field. So this is a generalized schematic of all that variability for this one specific protocol. So up here we have all the different small molecules that are used to activate and inhibit the Wnt pathways. And then we also have the time course, that all of these are used throughout the field. And if everyone did the same exact thing, this would look like a step function, but it doesn't. And so even if we are just looking at 4 variables, which are the Wnt activator, inhibitor, and their time courses, over 60% of these protocols are completely unique. So there's a lot of variability within the field, and our goal as a center is to help guide the field to have a more standardized protocols and have higher reproducibility. In addition, all of those protocols I was just talking about are to only make ventricular cardiomyocytes. These are studied because that's where a lot of injury and disease affects the heart, since that's where blood gets pumped out into the body. But there are a lot of different subtypes of cardiomyocytes. So right here, we're just looking at the left ventricular cardiomyocytes. But if we start with cardiac progenitor cells, there's all these different pathways that can be activated or inhibited to make many different subtypes. And it's important that, or it's negative that we're missing those other subtypes, because there are genetic diseases that impact other parts of the heart. And if we're only making left ventricular cardiomyocytes, we lose out on the ability to study those. So being able to automate this process would allow us to develop these protocols and also fine-tune them for a more reproducible results. So the goals of working with the DAMP lab were to really decrease the hands-on time that we have as researchers, so that we can focus on experiments and analyzing results. So when I was at the peak of my PhD doing a lot of experiments, I was probably doing 2 to 3 hours of just media changes a day, which really takes away from experiments and learning things from our results. So we would really like to reduce that and also create more standardized and reproducible results through this differentiation process. And then lastly, once we are at that stage of having a really standardized method to differentiate these cells without the addition of human error, being able to fine tune some of those variables so we could figure out which protocol out of all of those 200 plus protocols in the field are actually the most beneficial. So the year 1 goal of this pilot project was to just be able to get the stem cells to be in sterile culture and successfully differentiate them into cardiomyocytes. So I believe this is the first time the DAMP lab was working with mammalian cells. I might be incorrect on that, but and in the future, now that we've done that, we would really love to scale up in the number of lines that we're using, and also be able to test out different protocols, maybe to get different cardiomyocyte subtypes, or to see and compare protocols directly. So Rita, who is leading this project, came to our lab and chatted us to see how we're physically putting our written protocol into action. And she identified 3 different blocks of our protocol. The first is just plating the stem cells. This takes a lot of time just because you have the passaging and the spinning down and all of that. But then there's about 20 days where we're inputting different medias and different small molecules, but we're just essentially doing a media change and or a PBS wash. So this protocol, even though our inputs are different, it's the same thing physically. And then at the end, we basically replate the cardiomyocytes. They're more sticky than other cell types, and put them on a new matrix. So that takes a little bit longer, about 30 minutes a plate. In our first iteration, we were using the OT2, and Rita wrote a script that can basically remove all the media, add PBS, and remove the PBS, and then add the media. So one limitation that we've had in this is just the amount of liquid that we could handle. So right now, for the first prototype, we were using about 1 milliliter at a time, where we're using 12 milliliters in total. But we're still successfully able to differentiate these cells and keep them happy. So I will get into that. So to do this, one thing that Rita noticed when we were doing our different media changes is that we rocked the plates a little bit to remove all the dead cells that occur during differentiation. So she created and engineered an adapter that had a 30-degree angle to mimic this, and wrote the scripts to basically dispense the media on top and remove it from the bottom so that we're washing the dead cells out. So one issue we had when we first started was that due to the airflow of the HEPA filter that was installed and the angle of the adapter, the top half of the culture, the cells were drying out and they weren't happy when we were having that cell death. So Rita and the damp lab were really swift to adapt to this and re-engineered a 5-degree adapter so that we can still have some of that washing moment, but then also minimize the amount of time that cells were in an air interface. And we were able then to successfully get the differentiation to work. So the iPSCs that we use have a genetic tag that creates green fluorescent protein when a cardiomyocyte protein called titin is expressed, which you can see here. And using this, we're able to use flow cytometry to see how many of our stem cells actually became cardiomyocytes. And as a center in the study that we're doing that looked at 300 papers, the average reported in publication differentiation purity is 85%. And we compared the automated differentiation to the manual differentiation and found that we got about over 90% for the manual and over 95% for the automated differentiation, showing that we had success in creating pretty pure cardiomyocytes, which was really exciting to see as we move forward with scaling this up. So in conclusion, Rita and the DAMP lab wrote 4 open access scripts to automate this. And the nice thing about this is if we wanted to try out all those other differentiation protocols I was talking about, we can use the same script, we just input different media. They installed a HEPA filter into the OT2, which wasn't previously there, and did a lot of adaptations to get it to work with our system, and had some hardware adaptations also to help minimize our cell death and keep those happy. And in the end, we're able to get a really good purity of cardiomyocytes from our differentiation protocol. And so next steps are, we would love to increase the liquid handling ability so that we can maybe do 12 milliliters or even just more than 1 milliliter at a time so that we could maybe get this automation time down from 5 minutes to 2 minutes. Also implement the replating protocol. So right now we're doing media changes with the cells, but maybe being able to get to do the liquid handling with the cells in the media. And then also be able to expand so that we can increase the amount of plates that we're doing at a time. Asynchronously, I might be doing 40 plates at a time. So being able to get up to 100 would be really cool for the center. And so this is supported by Celmet. Josh and Mike have helped out a lot within our lab, and the DAMP lab, especially Rita. So I'll take any questions.
For you, just automating the liquid handling parts, were there any other like going to incubator or verification parts that you're automating or?
Yeah, I think that would be interesting in the future. Right now for the pilot project, it was just the liquid handling. But I think working on like the replating protocol where we're lifting up those cells or maybe even freezing them down to store them for later after mass production is definitely something we're interested in.
How do you create the adapters?
Rita could answer that.
Were there any challenges in scripting with handling to
really like what is it? Yeah, like oh, and it's a font. I'm killing myself.
Yeah, we even like switched up the plates on her one time, and there was an adaptation that was needed for that because each brand's different.
Yeah. Seems like something we were talking about yesterday about little bits of script that could be useful to other people. When you tilt the plate and then you actually just pull that out, like just that code, that would be helpful instead of trying to dig it out of a large protocol.
I had another question about how you guys are thinking of doing more One of them. I went to every booth I could find. So right now, you can use those in conjunction. So you can have full volumes, but also larger.
Yeah, so one of our most popular options is 4-1. We also have a tilt module, which will tilt the cells to the side. Then you can take you can also see some.
Yeah.
And you can tilt them to certain degrees.
As long as we're getting into the details. My question is for like in both the Obertons and in Hamilton. You know, I know that we talked a lot about biosafety and not contaminating cells and COVID, that we're pretty good at, but not contaminating the cells. That was a totally different kind of contamination issue to think about when we switched over to these projects with actual living things. And so what are the plans, you know, at the companies to deal with, I think, the urging demand for we want the stuff back and we want it to be alive.
Yeah, so we have an SCAP offering, which is basically a HEPA that's integrated into the stock.
Yeah.
We also have a lot of books that put them in safety rooms.
This small thing that's over in the demo, that yeah, and I know the OT2, the Flex, is going to have that as well. And then I guess if you put it in a facility that's like a cleanish room, you know, not maybe a cost or a cost 10,000 higher. Yeah, it should be pretty good.
Yeah, I was more referring to Star.
Yeah. Oh, the big thing. Oh, the yeah. Well, so the larger one is more of a challenge, right?
Yeah.
Yeah.
But pretty common. 7 dozen. Yep, we have advantage of their inputs. We work with Big Meat, which is real right in. Move plastics, all safety rebuilt, plates.
We basically make them.
A lot.
On the Emerald site, they have a little video that you can watch of their, and they have like everything in there is vented out either chemically or I'm assuming with from I think you can't tell from the video, but everything is vented in some way through the ceiling, and that's like a capital expense, right? So you're kind of like playing like, are you gonna make it into a capital expense or
I had to build a system before that.
yeah.
Yeah.
Yeah. We're gonna have
Very limited budget, you know. Just took regular orange shell, plug in,
yeah.
put them on wheels. If you have a very mild transverse flow across the neighborhood, we have all these going. You know, we put petri dishes out for a month, nothing ever breathed. So it's a cheap and cheerful. But it has a lot of bleach and bug.
Yeah. Yeah, because you can imagine if you
all come and get delivered.
because if you're doing like a 20-day culture,
Yeah,
and
an
on like
expensive
day
culture. The
17,
media is
it's like-
expensive. And we don't use any antibiotics at all.
It's amazing. Well, thank you.